All right, today we're going to look at Chapter 15, Part B, and we'll start with the thyroid gland. The thyroid gland is definitely butterfly-shaped. It has two lobes, and those lobes are connected by a median mass or a piece of tissue called the isthmus. If we were to look closer at the thyroid gland, we would see that it has follicular cells, and inside or within these follicular cells is an open space, a hollow sphere. So these epithelial follicular cells surround this hollow sphere, and this hollow sphere doesn't remain hollow. It actually gets filled with a glycoprotein called thyroglobulin, which is actually produced by those follicular cells. Colloid is essentially what we're going to call that fluid, but only when the thyroglobulin is mixed with iodine before they have been kind of combined to create thyroid hormone. So colloid is the fluid in the follicular lumen. It contains thyroglobulin and iodine, not processed yet to create thyroid hormone. In between the follicles of the thyroid, we would find other groups of cells called parafollicular cells. Para is a prefix that means nearby or close to, sometimes surrounding, but these parafollicular cells produce a hormone called calcitonin. So we'll look closer at thyroid hormone. There's actually two derivatives. But if we wanted to list the hormones that were made by the thyroid gland, we'd have to list both thyroid hormones. I'll go ahead and list them for you, T3 and T4, and calcitonin. Calcitonin is oftentimes overlooked, but it is produced by the thyroid gland. So here's a picture of the thyroid gland. Let me outline it with my cursor. We can see it has 1, 2 lateral lobes, highly vascularized, meaning it has a great blood supply, and it has a piece of tissue that connects the two lobes. That's called the isthmus. It sits below the voice box or the larynx. So here you can actually see the voice box, above which, is the hyoid bone. And so that gives you a general location for the thyroid gland, but also note that it sits anterior to the voice box, not just below it. We're also going to be able to find, if we looked on the back of it, the parathyroid glands. There's that para prefix again. So parathyroid glands means that they're somewhere near the thyroid gland, and we'll see that. But let's kind of stay focused on the thyroid gland. So here is a histology micrograph of the thyroid gland. So keep in mind this is two-dimensional, but we would have three-dimensional spheres or spaces filled with colloids surrounded by follicular cells. So we're seeing a two-dimensional image of that here. So here's one follicle. All these cells are follicular cells. Here's another follicle up here. There's multiple in this picture. These follicular cells surround a space filled with colloid. We know that's the precursor to thyroid hormone. And we can see in between two follicles are another group of cells, a little indistinguishable from a follicular cell except for location, but these parafollicular cells are the ones that secrete the other hormone of the thyroid gland that's not considered a thyroid hormone, and that hormone was calcitonin. So let's look at the thyroid hormones. I keep mentioning them. They're the major hormone produced by the thyroid gland. Thyroid hormone can be abbreviated TH. Oftentimes we group the two forms, thyroxine and triiodothyronine, together. The abbreviations for those two hormones are T4 for thyroxine and T3 for triiodothyronine. And we can convert T4 to T3 at tissue level because T3 actually binds better to peripheral tissues than T4. But T4 is definitely the major form that is made. It consists of 2 tyrosine, that's where we're getting T from, 2 tyrosine molecules with 4 bound iodine atoms. So tyrosine is a part of thyroglobulin. So if we cleave that off of thyroglobulin, then add the iodine atoms that exist in colloid, we can form T4 if we do 4 bound iodine to 2 tyrosine molecules. T3 also has 2 tyrosine molecules, and you probably guessed it, not as many iodines. It has 3 iodines bound to this part of colloid from thyroglobulin. So now we've put our T3 and T4 together, and so we've made our thyroid hormone. Again, T3 binds better to tissues, but T4 is made in a bigger abundance, so we can convert T4 to T3 at peripheral tissues. But both are iodine-containing amine hormones, meaning they're really should be water soluble, but if you remember back to part A of the endocrine PowerPoint, thyroid hormone doesn't really act like an amino acid hormone. It actually acts much more like a lipid soluble or steroid-based hormone. But keep in mind, it is an amino-based hormone. It acts like the steroids, so it's actually lipid soluble. So thyroid hormone is the major metabolic hormone. If you remember nothing else about the thyroid gland and thyroid hormone, remember what the thyroid gland produces, and that thyroid hormone specifically, both derivatives, are the major metabolic hormone. They have target cells all over the place in the human body. Thyroid hormone actually affects virtually every cell in the body. It enters target cells, and that lines up with acting like a lipid-soluble hormone for sure. And it binds, therefore, to intracellular receptors. We know that our receptor hormone complexes then go into the nucleus and are able to trigger the transcription of various metabolic genes. So again, thyroid hormone acting very much like a lipid-soluble hormone, even though it's made up of amino acids. So the effects of thyroid hormone, again, it's a major metabolic hormone, is generally going to be calorogenic. It's going to make cells increase their basal metabolic rate. Oftentimes, this produces as a byproduct of ATP production, heat. So a lot of the effects of thyroid hormone are calorogenic. So it regulates tissue growth and development. So we see definitely skeletal growth, nervous system development, reproductive capabilities, all relying on either permissiveness or aid that's provided by thyroid hormone in its actions. It's even able to maintain blood pressure. It increases adrenergic receptors in blood vessels. If you don't remember from AMP1, adrenergic receptors will bind to epinephrine or norepinephrine. So let's talk about how are we making thyroid hormone. Unlike other glands, thyroid hormone is actually stored in the spaces where we find colloid. So thyroid glands store hormone extracellularly, and this doesn't happen a lot for endocrine glands. And we're going to see that it resides in the follicle lumen. So we have that precursor, that colloid, and then we can generate thyroid hormone when needed. There's actually 7 steps to the synthesis of thyroid hormone. And it would only be the release of thyroid stimulating hormone from the anterior pituitary that causes the release of the stored thyroid hormone and then generates more synthesis in order to restock our stored supplies. So the first step in generating thyroid hormone would be to break down thyroglobulin. So thyroglobulin does contain tyrosine, but it's a glycoprotein. So we would need to first synthesize thyroglobulin using the Golgi apparatus, and then send it into the follicle lumen, or that space where all the colloid's stored. Then we need to get iodide from the blood, transfer it or transport it across the cell, and then send it into the follicle lumen. We would also then need to oxidize iodide to iodine. So we break it down some, kind of change it, move around its electrons. So what we've done thus far, in order to eventually break down thyroglobulin and get our tyrosines, is first make thyroglobulin, send it to the follicle lumen, trap iodide, send it to the follicle lumen, and then turn it into iodine. What we're going to do next is attach our iodine, so that's our oxidized iodide, to the tyrosines from the thyroglobulin. Now, this is mediated by a couple of enzymes. These enzymes are peroxidases, and we can create a couple of different things by combining tyrosine and iodine. We can create monoiodotyrosine and diiodotyrosine. That one always gets me. The abbreviation is much easier. So MIT for monoiodotyrosine and DIT for diiodotyrosine. Better the second time. And what this is tyrosine bound to 1 or 2 iodines. So MIT is formed if only 1 iodine attaches to a tyrosine. DIT is formed if 2 iodines attach to a tyrosine. And then if we link the MIT and DITs together, we can form our T3 and T4. Remember, T3 and T4 both have 2 tyrosines, and the number after the T tells you how many iodines they have. So if we link an MIT and a DIT together, we can form T3, whereas if we link 2 DITs, we can form T4. So once we form T4 and T3, we're going to endocytose a piece of that colloid, meaning the follicular cells are going to form a vesicle around some of this colloid and take the fluid into itself. But then we have to combine what's in the colloid that's not T3 and T4 with a lysosome so that the lysosome can kind of break down everything that's not T3 and T4, and therefore we'll have just the hormones T3 and T4 secreted into the bloodstream. So again, the most abundant secretion for the thyroid gland is going to be T4 over T3. So it's mostly T4 secreted, but T3 is also secreted, just not in as much abundance. But T4 therefore has to be converted to T3 at the tissue level because T3 binds 10 times better than T4 on peripheral tissues. So here are those 7 steps. I'm going to abbreviate them, make them a lot shorter since we just went through them. But let's start at step 1. We're going to make thyroglobulin. It's going to be transferred from the rough endoplasmic reticulum to the Golgi apparatus, and then finally to the follicle lumen. We're looking at one follicle cell, but there would be many around this follicle lumen space where the colloid is. So we made thyroglobulin and we transported it to the follicle lumen. We got iodide from the blood, we transported it into the cell, across the cell, out of the cell, into the follicle lumen. Next, we're going to turn iodide into iodine. So we'll oxidize iodide. Then we will form DIT and MIT by breaking down thyroglobulin and putting those tyrosines from thyroglobulin onto our oxidized iodide, which is now iodine. So we have now DIT and MIT in the thyroglobulin colloid, and then we need to endocytose these two substances. While doing so, they will combine to form T3 and T4. The vesicle form that contains thyroglobulin and excess materials plus T3 and T4 must combine with a lysosome in order to break down everything that's not T3 and T4. Once that occurs, we will have just pure T3 and T4 in the vesicle, and that can be secreted then into the interstitial space and cross over into the blood. So T3 and T4 are hormones. They travel in the blood. They behave a lot like lipid-soluble hormones, even though they're made up of amino acids. Therefore, their action once they're in their target cell is going to be direct gene activation, and the receptors are intracellular. So transport and regulation of thyroid hormone. We know it's transported to the blood, but T4 and T3, since they act like lipid-soluble hormones, must bind to helpful plasma proteins in order to be transported. So T4 and T3 are transported by thyroxine binding globulins, or TBGs. They both bind to, they both bind to target receptors, but again, T3 is 10 times more active than T4, so a lot of T4 is converted to T3 at those peripheral tissues. This does require an enzyme, but it's an enzyme that often is present at peripheral tissues. And the enzyme generally removes one iodine, and that makes sense since we're moving from T4 to T3. Thyroid hormone release is regulated by negative feedback, and if this is looking familiar, we did touch on this in Part A of the Chapter 15 PowerPoint. So this should be a review, but falling thyroid hormone levels would stimulate the thyroid-stimulating hormone to produce or be produced by the anterior pituitary, and then once thyroid hormone levels have risen, there'd be negative feedback on the anterior pituitary and sometimes also the hypothalamus to prevent further production of thyroid hormone and thyroid-stimulating hormone. We do know thyroid-stimulating hormone can be inhibited by growth hormone inhibiting hormone, dopamine, and increased levels of cortisol and iodide. And it's the thyrotropin releasing hormone, or TRH, that starts everything in this cascade is sometimes called the HPT axis. So here should be a familiar pathway. We've looked at thyroid hormone secretion before because we've looked at thyrotropin releasing hormone and thyroid stimulating hormone. We just haven't looked at thyroid hormone in detail, but now we know information about the entire pathway. So the hypothalamus is going to secrete releasing or inhibiting hormones. In this case, it releases thyrotropin releasing hormone. Thyrotropin releasing hormone travels in the blood to the anterior pituitary and causes the release of thyroid stimulating hormone. Thyroid stimulating hormone, its name tells you exactly what it's going to do. It's going to go to the thyroid gland and stimulate the production of hormones there. So it's definitely a tropic hormone. And then when it gets to the thyroid gland, it does cause the production of thyroid hormones, but this increase in the blood levels of thyroid hormones can negatively feed back on the anterior pituitary and hypothalamus to shut down this pathway. Thyroid hormones themselves act on target cells, which are most cells of the body. Hence, we've been saying peripheral tissues, but not any specifically. And what thyroid hormone is going to do is increase the metabolism of cells. A lot of the effects of thyroid hormone is calorogenic. So here's a great table, table 15.4, that summarizes kind of the effects of thyroid hormone, whether it's T3 or T4, so thyroxine or triiodothyronine. We're going to see increases in basal metabolic rate for almost all cells when acted upon by T4 or T3. We get a lot of calorogenic effects. We mobilize fats, glucose, we break down glycogen. We're going to see that the development of muscle, bone, and the reproductive organs relies, at least in some part, on thyroid hormone. So a great review, we can see also in this table how many systems are affected or rely on thyroid hormone. Let's also look at some homeostatic imbalances associated with thyroid hormone. The hyposecretion or kind of reduced secretion of thyroid hormone from the thyroid gland in adults can lead to a mucus swelling. So that's called myxedema. Symptoms include a low metabolic rate. Makes sense because thyroid hormone is our major metabolic hormone. So not having enough of it can lead to a lower metabolic rate, but it also can lead to thick and or dry skin, puffy eyes, feeling chilled, constipation, edema, mental sluggishness, lethargy, kind of symptoms you would associate with your cells not working at their optimal power or energy level. But the myxedema can be caused due to a lack of iodine. If this is the case, then myxedema actually develops a goiter. This is when the follicle cells overproduce thyroglobulin, but they're not able to make thyroid hormone. But the lack of iodine is kind of not really a problem for the thyroid gland in producing thyroglobulin, but it is a problem to getting thyroglobulin or the tyrosines out of the thyroid follicle lumen. So if we're not able to get thyroglobulin out of the follicle lumen, the thyroid enlarges, it's filled with more and more colloid, the follicles swell, and this is generally due to the fact that a lack of iodine decreases thyroid hormone levels, which is usually a trigger for thyroid stimulating hormone to be released and telling the thyroid, hey, we need more thyroid hormone. But in this case, we don't have iodine to complete the synthesis of thyroid hormone, so the thyroglobulin just keeps getting dumped into those follicle lumens and the thyroid enlarges. So thyroid hormone is critical for normal growth and brain development in early childhood. Again, it's the major metabolic hormone. There is a condition called congenital hypothyroidism, and it is usually caused by poor development of the thyroid gland. So thyroid hormone replacement is usually crucial and usually lifelong in these individuals, and this type of congenital disorder could be caused by pituitary problems, so damage to the pituitary, a tumor in the pituitary, or maternal medications that sometimes can affect the baby's ability to make thyroid hormone. Sometimes this symptom or condition is asymptomatic. Other things that may present are weak cry, poor feeding, constipation, prolonged jaundice. And if we go back, some of those symptoms sound or are very similar to the symptoms of hyposecretion of thyroid hormone in adults. Others are unique to the congenital hypothyroidism. Here in figure 15.10a, we can see a goiter. So that is a mucus swelling caused by a lack of iodine, caused by the overproduction of thyroglobulin, and the swelling of the thyroid gland. Another homeostatic imbalance to acknowledge is an autoimmune disease called a Graves' disease. And this is when the body makes abnormal antibodies. And antibodies don't normally cause hormone production, but in this autoimmune disease, the abnormal antibodies are directed against the thyroid follicular cells. And the antibodies actually act like thyroid stimulating hormones, so therefore, in someone who has Graves' disease, their thyroid gland is overstimulated. And so it produces a lot of thyroid hormone, and this gets dumped into the blood. So at first, a person who has Graves' disease, when they first develop this disorder, they may feel great. They're going to have a lot of energy. They don't run out of energy. They don't even need a lot of sleep. They get a lot done. They're very productive because they have this elevated metabolic rate. But eventually, this elevated metabolic rate starts to take its toll on the body. It can cause sweating, rapid and irregular heartbeats, nervousness, weight loss, even if the person is eating an adequate amount of food. One of the most kind of astonishing symptoms, or probably the most concerning, is exophthalmos. This is going to be protruding eyes or bulging eyes, and it can result, not always, but it's when the eyes protrude because tissue behind the eyes is either being used for nutrients, so breakdown of fat, or muscle in order to provide the energy for this elevated metabolic rate of cells, or it's because that tissue behind the eyes is becoming swollen or edematous and fibrous. The treatment for Graves' disease is generally surgical removal of the thyroid or treating the thyroid gland with radioactive iodine in order to destroy the kind of overactive thyroid cells. But it's usually a lifelong process of managing the symptoms. And here is an example in figure 15.10b of the bulging eyes, which are characteristic of kind of severe Graves' disease. Keep in mind that Graves' disease is an autoimmune disorder. Calcitonin is the hormone we're going to look at next, and we're still looking at the thyroid gland. So again, don't forget that calcitonin is produced by the thyroid gland, but it's produced not by the follicular cells, it's actually produced by the parafollicular cells, which are sometimes called C cells, which may be easier to remember because calcitonin, C cells. But parafollicular cells are found in between the follicles of the thyroid gland, and calcitonin produced by these cells is produced in response to high calcium levels, because what calcitonin does is it tones down the amount of calcium in the blood. It's going to decrease calcium levels. So if your stimulus is high calcium levels, that will be the trigger for the parafollicular cells of the thyroid gland to produce calcitonin, dump it into the blood. It acts as an antagonistic pair or part of that pair to parathyroid hormone, which does the opposite. Whereas calcitonin decreases calcium levels in the blood, parathyroid hormone is going to increase calcium levels in the blood. Now, calcitonin isn't really used a lot in the human body. It does exist, but it's not the number one most responsible hormone for calcium homeostasis. That would be parathyroid hormone. There's really no known physiological role in humans at normal doses. At normal physiological levels, calcitonin, not really important. We know what it does because we've studied it at higher than normal doses. And at higher than normal doses, it does the opposite of parathyroid hormone. But parathyroid hormone works just fine all on its own. But at higher than normal doses, calcitonin does inhibit osteoclast. Therefore, it inhibits or stops bone resorption and prevents release of calcium from the calcium phosphate salts that are in bone matrix. So we're not kind of eating up that calcium bone matrix and releasing calcium into the blood if we're inhibiting osteoclast activity. We're also going to see that at higher than normal doses, calcitonin can stimulate calcium uptake and incorporation into bone matrix. So when we do absorb calcium from, say, our small intestines, we're going to see if calcitonin has been produced at higher than normal doses, it would lead to that calcium being deposited into the bone matrix. So we're just basically putting calcium phosphate in the bone matrix or keeping it there because we're inhibiting osteoclast activity. So we're going to prevent adding more calcium to the blood. And over a period of time, this should reduce the amount of calcium in the blood. Hence, calcitonin toning down the amount of calcium in the blood. Again, not a huge physiological role because it's not produced in these higher than normal doses. So let's look at figure 15.8b again. So we have our follicular cells surrounding the colloid-filled follicles that had to do with thyroid hormone, and our parafollicular cells are in between those follicles. Again, they're the ones secreting calcitonin. All right, before we move on to the parathyroid gland, let's just kind of abbreviate what we talked about with the thyroid gland. So we know where it's located. It's anterior to the trachea, below the larynx. It has two lobes connected by a piece of tissue called the isthmus. It has a great blood supply and produces three hormones, T3, T4, and calcitonin. The follicular cells produce T3 and T4, but through a series of many steps that involve the production of thyroglobulin and the transport of iodide into the follicle lumen, and then we have to oxidize that into iodine. And we combine everything, then we transport it out, and eventually we'll get pure T3 and T4, and it'll go into the blood, but only when triggered by thyroid stimulating hormone. We looked at calcitonin. Calcitonin is going to be able to decrease the amount of calcium in the blood, whereas our thyroid hormones were the major metabolic hormone. So moving on to the parathyroid gland. So moving away from the thyroid gland, but we're still in the general area because these 4 to 8 tiny yellow-brown glands are embedded on the dorsal side or posterior aspect of the thyroid. They are produced by parathyroid cells. We do know that the parathyroid gland also contains oxyphil cells. So the parathyroid cells are going to produce the parathyroid hormone, and it's going to be the most important hormone in calcium homeostasis. So yes, calcitonin is going to be an antagonist, but it's mostly parathyroid hormone regulating our calcium concentration in our blood. And we see that parathyroid hormone is secreted in response to low blood calcium levels. That would be the stimulus for parathyroid hormone secretion. So the secretion of parathyroid hormone is responding to a humoral trigger. So that's a change of ions in the blood or lymph. But we would stop parathyroid hormone secretion once we have rising levels of calcium. So it would be inhibited by rising levels of calcium, that parathyroid hormone production. Parathyroid hormone can also be abbreviated PTH. And the target organs for this hormone are the skeleton, the kidneys, and the intestine. And we're going to talk about why. So let's look at where it's located. So here is one lateral lobe of the thyroid, another lateral lobe of the thyroid gland. On the back, so we have a dorsal or posterior view right now, we have these 4 little yellow kind of pea-shaped glands, and all 4 of these glands are parathyroid glands. Again, the parathyroid gland produces parathyroid hormone. It has the opposite effect of calcitonin, so we're not going to inhibit osteoclast activity like we did with calcitonin. In this case, the parathyroid glands, when they produce their hormone, parathyroid hormone, are going to stimulate osteoclasts to digest bone, to release the calcium phosphate from the matrix. And that will release that calcium to the blood and increase blood concentration of calcium. Other things we're going to see possible via the action of parathyroid hormone is the enhancement of the reabsorption of calcium from the small intestine, and we're also going to enhance the reabsorption of calcium from the kidneys. And this also can lead to the secretion of phosphate in the case of the kidneys. So we'll reabsorb calcium but get rid of phosphate. But phosphate is going to be present in the blood due to the stimulation of osteoclast activity. So it helps to get rid of this excess. We're going to be able to increase calcium reabsorption via the small intestine by promoting the activation of vitamin D by the kidneys. So we do need activated vitamin D or calcitriol in order to absorb the maximum amount of calcium via our intestines. So through that intestinal mucosa. I highly recommend figure 15.12 when you're looking at the effects of parathyroid hormone on the target organs, bone, the kidneys, and the intestines. It's a great figure for showing us kind of what happens when our stimulus is hypocalcemia or a decrease in blood calcium levels. And what we're going to see is hypocalcemia as a humoral trigger will cause the parathyroid glands to produce parathyroid hormone. And that hormone will go to the blood and act on target tissues, the bone, the kidneys, and the intestinal mucosa. It'll cause calcium to be released by or from the bone matrix of the bone because we're increasing osteoclast activity. It'll tell the kidneys to reabsorb more calcium. So this is going to affect the kidney tubule system. And we're going to increase the activation of vitamin D by the kidneys. So the kidneys are involved in the activation of vitamin D. And if we have enough vitamin D, we can increase calcium absorption through the intestinal mucosa. Most absorption of nutrients does occur in the small intestine. That's why we're seeing the absorption of calcium as a nutrient in the small intestine as well. All of these things, when acted upon by parathyroid hormone, work together to produce more calcium that ends up in the blood. So if we start out with our initial trigger, hypocalcemia or low blood calcium, our final result, as all of these target tissues work and are acted upon by parathyroid hormone, would be an increase in calcium levels in the blood. Let's look at some homeostatic imbalances. So hyperparathyroidism, that's going to be extra parathyroid hormone being produced, but that could be due to a parathyroid gland tumor. And that would cause extra parathyroid hormone, so that would cause extra calcium to be leached from or to leave bones. It could depress the nervous system because calcium is important for things like neurotransmitter release. Calcium is even important for things like mitosis. So changing calcium levels can definitely have an effect on organ systems. An example of severe hyperparathyroidism is osteoitis fibrosa cystica. Again, a severe form of hyperparathyroidism resulting in easily fractured bones because so much calcium would be leached in this severe form, and that would cause them to soften and deform, become very brittle. Hypoparathyroidism is usually something that would follow after something happened to the parathyroid glands, maybe after injury, or if we had to remove one of them because of a tumor. This could lead to a decrease in the amount of calcium in the blood because of a lack of parathyroid hormone being produced following gland trauma or removal. This could also interrupt functions of the nervous system, the respiratory system, and could even lead to death because it can result in tetany, respiratory paralysis, and respiratory paralysis can lead to death. And that's going to be it for the first part of Part B for the endocrine system. Next, we're going to look at the adrenal gland, so stay tuned for that.